Alrighty, so today we're going to talk about the cell cycle. So, the cell cycle is essentially the life cycle of a cell. It will go in between these different phases in a cyclic nature, so it'll continue from one to the next, to the next, to the next, and come back and go again. Specifically, when we talk about the cell cycle, we're looking at the time from one division to the next, because sometimes a cell needs to make new cells. You don't have a finite number of cells in your body. They are continuing to grow, divide, make new ones, recycle, get rid of some. So it is a constant flux with the cells. But sometimes it is necessary to make new ones. When they do this, they will copy it and they will divide. And what you end up with are two daughter cells. That's what we call the two cells that come after a cell division. The one that started, generally, we call it the parent cell. But the daughter cells are the ones that come after. When we talk about the phases, we can kind of break it down into 5 general phases. We have G1, and the G stands for gap. The S phase stands for synthesis, and in a minute all this will make sense. G2 is gap 2. Then we have mitosis. Mitosis here, folks, for this particular segment, we're just going to talk about it a little bit, and then next segment we'll go much more in depth on mitosis and the actual phases. But mitosis, this is where we're actually doing the dividing of the cells. In all of this stuff here, we're not dividing. The cell is living its life and doing its thing. And then cytokinesis, which is essentially just that finite point where, yay, we have a split, and now we have our remaining 2 cells. These 2 cells, those are our daughter cells that we have. So let's take a look at what that looks like. So here, this here is our G1. This is our S. This is our G2. Notice this term here, interphase, right? So all of these 3 phases here, our G1, our S, and our G2, this is all part of interphase. Inter meaning between, and that's between cell divisions. So the time in between cell divisions is called interphase. In G1 here, this is where a cell is growing. DNA replication is occurring in our S phase. Remember, synthesis, right? We're making new DNA. And then here in G2, we're prepping. We're getting ready. G2 is right before mitosis. Notice we have mitosis over here. So we're getting ready for that during G2. And then here are our phases. You might remember them: prophase, metaphase, anaphase, and telophase, or PMAT. May sound familiar. And then cytokinesis, that's that final point of cell division where now we can start the cycle again when we have 2 daughter cells that result, and they can both cycle back into the cell cycle. So another nice little visual here. Here's our G1 where our cell is growing. It will physically get larger, develop more structures. Then we reach this point here where essentially the cell can decide whether it's going to keep going through the cycle or spit off this way. We'll talk about that in a minute, but that's the R point. Here is S, synthesis. Right? We're making another copy of the DNA. Notice how many chromosomes we have here. 1, 2. And notice over here we have 2 copies of that one on top and 2 copies of that one on the bottom. We have replicated our DNA. Then we go in here into G2. G2, gap 2. This is where there's a gap in the replication and it's a gap in the division. So it's where the cell is just doing its job. It's just doing stuff that the cell needs to do to be a cell. And one of those things it needs to do to be a cell is often divide. So here we're prepping during our G2, And then here is our point of division. Here is mitosis. Notice we've got one cell that looks like it's busting out into two, and then these two cells will individually go off and re-begin their cycle and move into cell cycle and pop into G1 or gap 1. So interphase, right? Interphase, this is the longest phase of the cell cycle. The cell spends the most time here in interphase, and that's because this is when the cell is doing its job. It does whatever it is that cell does. If it's a liver cell, if it's a cell in your lungs, in your skin, in your muscles, whatever, it's doing its job. It's not necessarily, well, it might be worrying about dividing if it's in gap 2, but it's not dividing. This is the in-between, interphase, in between these divisions. At this point during interphase, we're not going to see chromosomes. You know those little X's that they showed in the previous pictures? We're not going to see chromosomes. We're going to see them in what's called chromatin. So what we'll see is this big mess, this big spaghetti mess. That is chromatin. It's unwound DNA. So our 3 different phases, G1, S, and G2. In G1, we're prepping for DNA replication. It's doing its normal functions. It's doing its job. And we're getting ready for everything that we need to do. Go forward. Replication, right? S is for synthesis. And then in G2, prep for mitosis, because time-wise, this is right before we enter mitosis. Now, there is this special phase, which I'll bring up, but we don't have to go too in depth here. Called G0. And essentially what G0 is this is right at that R point where sometimes a cell will sort of press pause. It will be considered a state of arrest or inactivity. But what it does is it leaves the cycle for a minute. Some cells will stay here in G0, things like cardiac cells, nerve cells, red blood cells, they enter G0 and stay. But essentially the big picture here is in G0, they're not worried about dividing. Another point here, cancer cells cannot enter G0 because cancer is uncontrolled cell growth. So they continue to divide and divide and divide and divide, and they will keep going through the cycle uncontrolled. The thing that's neat, cell cycle rates differ between different cells. Not every cell can go through the cell cycle at the same rate. So an embryo cell cycle is 3 minutes. You're growing incredibly fast when you're an embryo because you are trying to go from one teeny tiny little cell to trillions of cells very, very quickly. Your esophagus, 2 to 3 days, intestines, 6 days. Your red blood cells have a life cycle of 120 days. Your red blood cells can last anywhere from 10 hours up to decades, depending on what they're doing. So even just within some of these different areas of your body, the cell cycle is so different rate-wise. And then the last little chunk here of the cell cycle is here's mitosis. This is our actual cell division. Oh my goodness. So messy. Oh, sorry guys. This is our actual cell division. So notice we've got G2 kind of pointed out here. PMAT. I'm going to erase this because prometaphase is kind of one of those things that we're getting really, really technical and we don't need to talk about that. So for our phases, we're just going to talk about PMAT. Prophase, metaphase, anaphase, telophase, and then cytokinesis. This is the final point at which we now have 2 daughter cells. So for now, we're just going to leave it at that. And when I see you on our next episode, we will talk more about PMAT, those phases of mitosis, and what that really means.